to let people in. There we go. They didn't really understand how they worked. So, what we are going to be looking at is the history of how it came to be this molecule that carries genetic information, how it was identified, and how to understand how the genes control things. So again, we understood that there were genes. We understood that there were these things that could control traits, but we did not understand how it did it. So again, and I always say this as a teacher, it's one thing to understand that something does happen. Understanding why it happened is a whole nother situation and a deeper level of understanding. Okay. Um, we are now on the third bullet point. The discovery of the chemical nature of the gene began in 1928. Now, some people can argue that this actually started much earlier than this, but that's okay. The guy that really begins to do work on understanding the nature of genes is 1928, Frederick Griffin. And basically, 1928, Frederick Griffin is trying to understand certain types of bacteria that produce pneumonia. For some historical context, please understand that this is about 10 years after, a little more than 10, a little less than 10 years actually, after the Spanish flu pandemic, which was the last large pandemic that swept through the earth and killed off a bunch of people. So were looking to understand. Griffin was specifically trying to understand how pneumonia, which was the Spanish flu, why it was and how it was that it was able to produce bacteria that creates pneumonia and essentially kills people. So, again, what we're looking to do here is understand how Griffin's experiment um, led to, if you will, understanding of DNA, because they sort of seem like two separate events, right? Like we're looking at understanding pneumonia. How does bacteria produce pneumonia? And we end up discovering a fact about DNA. And that's kind of the beautiful thing about science. Oftentimes, things that don't seem to be related are, and sometimes things that totally we think are related aren't. All right, so let's move on to Griffith's experiment. Okay, so again, Griffin was trying to understand pneumonia, and he isolated two different strains of the same bacterial species. Okay, both strains grew extremely well in culture, and but only one of them was disease causing. Okay, so we call them S and R strain. The S strain was called S strain because the cell membrane of that bacteria seemed very, very smooth. The harmless bacteria was what we call the R strain, and the R strain was considered rough because when you looked at it under a microscope, the outer cell membrane, outer cell wall of this bacteria had a rough texture to it. So nothing, monumental. This is like one of those times where naming in science actually makes sense. S strain stands for the smooth strain because it had a smooth outer edge. R strain was the rough strain. But the most important thing here to remember is that the S strain was deadly and the R strain wasn't. But they were both the same bacterial species. One caused pneumonia and was deadly, smooth, and the other one basically was pretty harmless. I mean, keep in mind, right now, every single one of us has a ton of bacteria in our bodies. And the vast majority of the bacteria that live in our bodies are actually not harmful at all. They're mostly pretty, actually helpful. Anyways, moving on. So, the disease causing bacteria, like we said, was the S strain. Um, Grew into smooth colonies on culture planes. And like we said before, the harmless bacteria, the R strain, produced colonies with very, very rough edges. So, not exactly the most exciting naming factor. Please, if you need to say something, you need to put it into the, you need to unmute and say something. I cannot see the chat when I'm sharing my screen. I can see that there's something in the chat, but I can't actually see the chat. It just popped, like, I don't know if you guys can see it right now, but I just get like a little notification that somebody's trying to chat with me, but I cannot actually see the chat. So if there's a question, please unmute and ask. All right. So again, disease causing bacteria, smooth, harmless bacteria, rough. But again, these are the same kinds of bacteria, but just like one's deadly, one's not. Okay, so here is the actual experiment. If he took mice, because he did his experiment on mice, and he injected them with the disease causing bacteria, the mice developed pneumonia and died. Not a surprise, like at all. Like, that totally makes sense. If he injected the mice with the harmless bacteria, then the mice stayed healthy. I don't imagine that surprises anybody either. Okay. So again, I put the deadly bacteria into the mouse dies. I put the harmless bacteria into the mouse stays healthy. So, Griffin wanted to figure out what the difference was between these guys because they were the same type of bacteria, but what caused the S strain to produce a toxin that made the mice sick? Okay, so that's what he was trying to understand. What was it that was making this bacteria different that killed off the mice? And that's what he was seeking to understand. All right, so first he takes a culture um, of the S strain and he injects it into, and he heats it up. And when he heats it up, he kills off the bacteria. So then he kills the bacteria and he then injects it into the mice. Okay, awesome. That being said, big surprise, the mice survived. So when the mice survived, it suggested that the cause of pneumonia was not some kind of toxic thing from these disease causing bacteria. It must have been something about the life process of those bacteria. Because when they were dead and their life processes were stopped, they no longer killed the mice. Okay. Again, that sort of seems a little obvious to us. And that's okay. Trying to figure out where we are in the notes exactly because my notes opened up funny. Okay, so what happened next? So, what he did next was he mixed the dead heat killed bacteria with the harmless bacteria that were still alive. And he then injected the mice and they died. So, in other words, something about a dead bacteria, which used to be harmful, mixing it with a harmless bacteria, something changed. Good morning. All right, so something had changed. So as we good morning, love. So as we mix these together, something changed to cause this to be deadly. So now Griffin wants to understand what caused it to change. So again, the question becomes how? How is it that the S strain is causing death even though they were dead? Again, guys, I'm seeing stuff in the chat. I can't see the chat. All I can see is that you put something in the chat. Like it basically comes up as like, hey, you have a chat. Kind of like it's like a notification. If there's something you need to ask, you need to unmute and ask it. I cannot see the chat. So if you need to talk to me, you actually need to unmute and talk to me. All right. So he said he called this process transformation. Because something had happened to the once harmless bacteria and had changed into something else. Okay? So, deadly bacteria kills mice. Harmless bacteria doesn't kill mice. If I heat kill the if I heat kill the deadly bacteria and I add it into some live bacteria, then that somehow is transformed and now I'm killing mice again. Something about these life processes is causing the transformation to happen. And something is being transferred, right? Because those harmless bacteria are obviously somehow picking up something from the deadly bacteria causing death to happen. All right. So, moving on to transformation. So, since the ability to cause disease was obviously inherited, and again, they understand inheritance because that comes from Mendel, and that was almost 100 years ago from this guy. Griffin concluded that the transforming factor had to be a gene. And in this picture, guys, you see the sum of the experiment. So basically, disease-causing bacteria causes a mouse to die. The harmless bacteria lets the mouse live. When we heat kill the bacteria, just the S strain by itself, the mouse lives. But if I mix the harmless and the heat killed, it somehow kills my mouse. He said that somehow this heat killed bacteria was causing a transformation to the harmless bacteria to cause death. And again, he reasoned that the transforming factor had to be some sort of gene or allele. All right, so you are now at the part of your notes that says molecular cause for transformation. All right, so fast forward. A group of scientists wants to figure out which molecule in the heat killed bacteria was most important to transformation. So remember, there are 4 molecules that all living things had. We talked about this last semester. There are carbohydrates, there are lipids, there are proteins, and there is DNA or nucleic acids. Those are our 4 macromolecules. So what they did was they added enzymes to destroy each of the different molecules. So when they destroyed the protein, transformation didn't occur. When they destroyed the carbohydrate with an enzyme, transformation did not occur. I'm sorry, I'm saying this backwards. When they destroyed the carbohydrates, transformation still happened, the mice still died. When they destroyed the lipids, transformation happened, mice still died. When they destroyed the proteins, transformation happened, mice still died. But when they destroyed the DNA, transformation did not happen, and the mice lived. Therefore, what they reasoned was that DNA has to be the transforming factor, because when you destroy DNA, transformation doesn't happen. This meant that DNA had to be the molecule carrying the genes, carrying the alleles, carrying these traits that Mendel had talked about years previously. We are now on the part of the notes that are, say, bacterial viruses. All right, so fast forward to I want to say Hershey and Chase were doing their work mostly in the 40s, maybe the 50s. I could be wrong. So several different scientists repeat the experiment. Hershey and Chase, so Alfred Hershey and Martha Chase, performed the most important experiment relating to the discovery of this. Hershey and Chase studied viruses. Now, keep in mind, viruses are non living particles that can infect cells. Viruses do not display all 7 characteristics of life that we've talked about previously. They are not made up of cells and therefore are considered to be non living. They cannot reproduce on their own, so they are considered to be non living. What's it called? As far as there was one other thing that they do that. There was one other character of life that viruses don't follow, and I can't remember what it is off the top of my head, but I'm sure it'll come to me. Anyways, Hershey and Chase studied viruses, and we know that viruses can infect living cells. So there was some controversy as to what the nature of DNA was, and a lot of people really thought that proteins were the ones controlling the genes. So what they do is they study using a bacteriophage. So a bacteriophage is a bacteria eater. Like literally, the word phage, I want to say it comes from Greek, and it means to eat. So the word bacteriophage literally means that these viruses eat bacteria. And what you see here in the picture is a typical bacteriophage. They have a head and a tail. The head has the viral DNA in it, and then the tail contains these sort of like little clamp looking legs, the tail fibers that will actually connect to your bacteria. So bacteriophage enter the bacteria. It will attach to the surface of a bacterial cell, and it injects its genetic information into the bacterial cell, and then it sort of hijacks the bacteria. Because the viral genes then produce many new bacteriophages, which destroy the bacteria. It splits open and hundreds of new bacteria burst out. Literally, it hijacks and rewrites the DNA of the bacteria, so the bacteria basically starts to code for creating new viruses as opposed to creating whatever the bacteria genuinely needs for life, which is kind of brilliant. From an evolutionary standpoint, but hey, that's just me admiring it. By the way, this is also why we see a lot of viruses change over time. Like we've seen this obviously with the coronavirus in our time, because pretty much it is just DNA. And if DNA gets copied wrong, it causes a mutation, and those mutations cause different variations of a virus, which is what we've seen happen here with COVID, right? We started with the original strain, and then there was a second strain, and then there was a Delta strain, and now we're on to the Omicron strain. And these happen because of the nature of what viruses are. They're basically a bunch of protein and DNA. And when that DNA doesn't copy correctly, it will cause a mutation. But that's just sort of a side note of stuff that I find interesting. All right, so the Hershey Chase experiment was actually kind of brilliant. What they figure out is that these bacteriophages are composed of DNA in the core and a protein coat or outer shell. And they want to figure out which part of the virus, is it the protein or the DNA core, that enters into the bacterial cell, causing the bacteria to die. So here is a quick little animation to show that. Hopefully, if it works.
In the last video, we began to see some pretty good evidence that DNA was a molecular basis for inheritance. And we saw that from the work of Avery, McCarthy, and MacLeod, where they tried to identify whether it was DNA or proteins that acted as the transformation principle in Griffith's experiments. And I encourage you to watch that video if all of this sounds unfamiliar. But even their work in 1944 was not viewed as conclusive evidence. It was viewed as strong evidence, but not conclusive evidence, because remember how they did it. They took the heat-killed smooth strain. The smooth strain, you might remember from Griffith's experiment, was the virulent one. They heat-killed it. When you heat-kill it and inject it in a mouse, it didn't do anything to the mouse. But if you took the killed heat, if you took the heat-killed smooth strain and put it with the rough strain, it somehow transformed the rough strain into the smooth strain, into the virulent strain. And so they took the heat-killed smooth strain and they took out its different components, and they eventually were able to isolate one that was able to transform the rough strain into the smooth strain by itself. And then they applied all sorts of chemical tests to it and said, hey, there's pretty good evidence that this is DNA. But it wasn't conclusive because, well, maybe they didn't purify it properly, or maybe there was still a little bit of protein. Maybe it was mostly DNA, but maybe it was a little bit of protein that was still left there that actually did the transformation. So the scientific community, they weren't just saying, hey, that looks pretty good, let's move on, let's just assume. They wanted to continue to test it, and especially test it in different ways. And the conclusive evidence didn't come until a few years later, until 1952, when Alfred Hershey and Martha Chase decided to study T2 bacteriophage. So let me write this down. T2 bacteriophage. This is phage that infects bacteria. Bacteriophage. When you hear the word phage, we're referring to viruses. Now they knew that T2 bacteriophage was composed of proteins and DNA. And they didn't, well, we now know that it's a protein shell and there's DNA inside. But they, from their point of view, they said, okay, it's made up. If we look at the stuff that this virus is made of, it's protein and DNA. So protein plus DNA. And they knew that this virus, when it infects bacteria, it injects something into that bacteria. So it injects something. And that something is what hijacks that bacteria's genetic information to start producing more of the T2 bacteriophage. So if they could identify the something that gets injected, if they could figure out if that something was either protein or virus, then they would have conclusively proven that if they could show that something was not protein or virus, if it was protein or DNA, if they could show that it was either protein or DNA, then they could show conclusively that it's either the protein or the DNA that forms the molecular basis. And so they were actually quite skeptical of Avery, McCarthy, and MacLeod's experiments. Hershey and Chase actually thought that they were going to show that it was the protein. And remember, this whole time people are like, protein, we know it's these complex molecules that have these different shapes and all these different amino acids. It seems like that's much more likely to encode the complexity of genetic information than DNA. They didn't have an appreciation for the structure of DNA at this time. So they devised an experiment to figure out what is that something that the T2 bacteriophage is injecting. Is that something protein? Is it protein or DNA? So this is the question. So what they do is they take they develop two batches of T2 bacteriophage. One batch of the T2 bacteriophage, they do it in the presence of radioactive phosphorus 32. In the other batch, they I guess you could say they grow that T2 bacteriophage in the process of another radioactive isotope, but this time it is of sulfur. This is sulfur 35. So why are they doing that? Well, phosphorus is found in DNA. So in this first batch, the radioactive marker, you could say, is going to incorporate itself into the DNA. In the second batch, sulfur is found in the protein and not in the DNA, and so this would actually tag the protein part. And if you're wondering, well, how do you develop these radioactive batches? Well, you let the viruses hijack cells in a medium that has either the radioactive sulfur or the radioactive phosphorus, and as they reproduce, they are going to incorporate that radioactive material into either the protein or the DNA of the new viruses that get produced. So anyway, they were able to produce some of the T2 bacteriophage in the presence of the radioactive phosphorus, and they knew that way that the DNA would get that radioactive material in it. And then with the radioactive sulfur, they said the protein would have that radioactive sulfur. And then for each of those batches, they then infected bacteriophage with them. And they said, okay, they're going to inject something into the bacteriophage. And to figure out what that something is that was injected into the bacteriophage, they take the products in either of the two scenarios. They first blend them up so that all of the stuff that's left outside gets taken off of the surface of the bacteria cells, and then they stick it into a centrifuge. And the centrifuge is, you can imagine, is kind of just a big spinning machine. If you were to take a test tube and take it sideways, one way to think about it, put it sideways like this, put maybe like a stopper in it so nothing leaks, and then you spin it around really, 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 really fast. What you're going to find, you can actually generate significant G forces. And so the heavier stuff is going to gravitate to the bottom of the test tube, or to the right, the way when it's on its side. And the lighter stuff is going to gravitate to the left. And it turns out that the bacteria, the actual bacterial cells, those are heavier. So the bacterial cells are going to go towards the bottom of the test tube, and they're going to form a material that we call the pellet. And then all of the other stuff, all of the fluid and the leftover phage parts, those are going to go up to the top of the test tube, and we call that the supernatant. Supernatant. I always have trouble pronouncing that. Supernatant. And so they said, look, if we look at the pellet, which they knew had the bacterial cells in there, or you can even say the remnants of the bacterial cells, if we look at if the pellet contains phosphorus, that means that the DNA, our radioactive DNA or our tagged DNA, made it into the bacteria. But if it contains sulfur, that means that the protein made it into the bacteria. And what they found is that the radioactive phosphorus was in the pellet, which allowed them to conclude that, hey, it's the DNA from the virus that made it inside of the bacteria and not the protein. And then they said, well, it must be. Wow, Avery, McCarty, and McLeod were correct. It's actually the DNA that is this transformation principle that can go in and hijack the machinery of the bacteria to produce more of the actual virus. So this is a really, 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 really big deal. Once again, we started with Mendel segregate and assort in certain ways. They seem to be discrete. Bover and Sutton said,
All right, so basically, what this guy just went through in a very, very long-winded way, I know he was kind of proud of him. He sort of reminded me of me. Is that at the end of the experiment, because of how it was concluded, they found without a doubt that the transforming principle in these bacteria that carried the gene that carried these traits had to be DNA. But again, this wasn't like an overnight thing. As always, um, these things take time, and it went through several different experiments. All right, so this pretty much just covers what he just talked about. I don't know where we are on time.